Welcome to the Deep Dive. Today we're diving into the world of Yersinia.
Specifically for you, the medical microbiologists out there.
That's right. We'll be focusing on how this information relates to your work.
We've got some really interesting research and notes compiled here.
Including, of course, insights from principles and practice of clinical bacteriology.
We'll be covering those three big pathogenic species, Yersinia pestis,
Yersinia pseudotuberculosis,
and Yersinia enterocolitica,
we're going to break down their characteristics.
their clinical significance.
And all the latest on diagnosis, treatment, and how to keep them under control.
You know, it's hard to talk about Yersinia without bringing up Y. pestis and its infamous role in history.
Oh, absolutely. The Black Death, right?
Yeah.
It really shows just how devastating these tiny organisms can be.
The Black Death was a major turning point in human history.
It's amazing to think how it shaped demographics and even cultural landscapes.
And what's really fascinating is that Y. pestis still poses a threat today.
Especially in certain parts of the world.
Exactly. It reminds us that even with modern medicine, these ancient foes are still around.
But before we get too dramatic, let's start with the basics.
Okay, so what are we dealing with when we talk about Yersinia?
Well, all three of those pathogenic Yersinia species are gram-negative coccobacilli.
And here's an interesting thing. They actually prefer cooler temperatures.
Really? So our cozy 37 degrees Celsius isn't their ideal environment?
Not really. They thrive best between 25 and 29 degrees Celsius.
So are they like the polar bears of the bacterial world?
You could say that. This preference for cooler temperatures is really important for their survival.
Especially for Y. pestis, which can live quite happily in fleas.
It makes sense. Fleas are like their personal AC units.
Speaking of Y. pestis, I learned something surprising.
Oh.
It's actually a clone derived from Y. pseudotuberculosis.
It's a pretty recent evolutionary event. It shows how dynamic bacterial evolution can be.
So in a relatively short time, Y. pestis evolved to cause this incredibly impactful disease.
Now that is an overachiever, even if it's a terrifying one.
It makes you wonder how these bacteria became so remarkably pathogenic, especially Y. pestis.
Well, each species has its own set of virulence factors.
But instead of getting lost in the details of each one, what's their overall strategy?
Especially, how does that strategy differ between Y. pestis and those enteropathogenic Yersiniae?
Okay, I'm all ears. What's the difference in their approach?
Think of it this way. For Y. pestis to be transmitted by fleas, it needs to cause a really widespread infection.
It needs to spread through the whole body.
Exactly. It has to be highly invasive and really good at evading the immune system.
Like a bacterial blitzkrieg.
You got it. But Y. pseudotuberculosis and Y. enterocolitica are different.
They don't need to be as aggressive.
Right. They can be transmitted even if the host survives.
So it's better for them if the host stays alive.
It actually is. If the host is alive, it can keep shedding the bacteria.
Okay, so it's more of a slow and steady wins the race tactic.
That's a great way to put it.
So two very different approaches, but both effective in their own way. So what are some of the key weapons in Y. pestis' arsenal that make it so potent?
Well, one of the most interesting things about Y. pestis is this unique plasmid it has, PFRA.
Another plasmid. What's so special about this one?
This one encodes the F1 antigen, and it's crucial for the bacteria to survive in fleas.
So it's not just about causing disease, it's manipulating the vector to help itself survive.
Exactly. And it also helps it spread to new hosts.
Wow, clever.
Yeah.
But I bet the PFRA plasmid has more tricks up its sleeve.
It does. The F1 antigen also creates a protective capsule around the bacteria.
So it's hiding from the host's immune system.
Exactly. It's a common strategy, but Y. pestis takes it to a whole new level.
So it's like wearing a disguise A&D, having a VIP pass to the flea party.
You got it. And that's just one aspect of Y. pestis' pathogenicity.
There's more.
There's another plasmid, PPST, and it has the gene for something called Pla protease.
And what does this Pla protease do?
Think of Pla as a getaway driver for the bacteria.
A getaway driver.
It activates a host protein called plasminogen, which helps Y. pestis spread from the initial infection site.
Oh, so it's like it's cutting a path for the bacteria to escape the immune system.
That's a great way to visualize it.
But wait, you said it was a multitasker.
Oh, right. Pla also breaks down complement, another part of the immune system.
Oh, right.
And it helps Y. pestis stick to laminin.
Laminin.
It's a protein found in our tissues.
So it's helping the bacteria get a foothold in the host.
Precisely.
Wow, it sounds like Y. pestis is really well equipped to survive and spread.
It's a formidable opponent, for sure.
Do the other Yersiniae have any unique tricks?
Well, Y. pestis and Y. pseudotuberculosis share something called the pH6 antigen.
And what does that do?
It's thought to be involved in immune evasion, maybe acting as a sort of stealth cloak.
So they're not just hiding, they're blending in.
Exactly.
And what about virulence factors shared by all three pathogenic Yersinia species?
All three of them share a remarkable virulence factor. The TYV plasmid, also called the LCR plasmid.
More plasmids. What's special about this one?
This one encodes a type 3 secretion system.
Okay, and in plain English?
It's like a tiny molecular syringe that injects proteins directly into host cells.
So they're injecting something into our cells?
Yes, these proteins, called YOPs, disrupt the host's immune response.
They're like shutting down the alarm system.
You could say that.
That's incredible.
And this lets Yersinia survive outside of host cells.
So they're playing a different game than those bacteria that invade and replicate inside cells.
Exactly. It's a unique strategy.
So what else does Y. pestis do to make us sick?
Well, it actually modifies its lipopolysaccharide, or LPS.
LPS.
It's a component of its outer membrane.
And it changes this.
It does, and this modification makes it less toxic to the host.
Wait, less toxic? Wouldn't it want to be as deadly as possible?
It's about survival and transmission.
So if it kills the host too quickly?
It won't have a chance to spread it.
So by being less toxic, it can prolong the infection and increase its chances of getting picked up by a flea.
Exactly. It's playing the long game.
It's like a parasite that needs its host to survive so it can spread.
And to suppress the immune system even more, Y. pestis uses a protein called LCRV,
So it's like it's calming down the immune system's alarm bells.
allowing it to multiply in the host.
But pestis has really thought of everything.
Now, let's not forget about a virulence factor shared by all three pathogenic species.
Okay, tell me more.
It's called Yersinia bactin.
And that is?
It's a cider-4.
A cider-4.
It lets these bacteria steal iron from the host.
Like a bacterial iron pirate.
A very accurate analogy.
Amazing.
The genes for Yersinia bactin are located on a high pathogenicity island on the chromosome.
So it could potentially be transferred between different bacteria.
Exactly, and that's a bit concerning from a public health standpoint.
Yeah, it could make other bacteria more pathogenic.
That's the worry.
So what about the enteropathogenic Yersinia, their own unique tools?
They have a few. Y. enterocolitica and Y. pseudotuberculosis have proteins called InV and ALE.
What do those do?
They allow them to invade epithelial cells.
The cells lining our intestines.
Exactly.
So they're getting inside the cells.
Yes, and InV and ALE are particularly important for Y. enterocolitica.
How so?
They bind to specific receptors on the host cells.
And
This allows them to attach and then slip inside.
sneaky. And what happens once they're inside?
They can cause all sorts of problems. Y. enterocolitica also makes heat-stable enterotoxins,
And those cause?
diarrhea,
Oh, no fun.
and it has something called MIF,
Which is?
a fibrillar adhesin.
Meaning?
It helps it stick to the intestinal lining.
They really are set on making us miserable.
It seems that way.
Yep.
So we've covered the microscopic world of these bacteria.
What's next?
Let's shift our focus to how these infections actually look in patients.
Okay.
Now let's shift gears from the tiny world of bacteria and see how Yersinia infections actually show up in patients.
It's crucial to know how they manifest, right? To diagnose and treat them properly.
Exactly. So let's start with the most famous one, plague, caused by Y. pestis.
Oh, yeah. I remember reading about those classic buboes in Principles and Practice of Clinical Bacteriology.
Yeah.
But what else should clinicians be looking for?
Well, the bubonic plague, the most common type, usually presents with those painful, swollen lymph nodes.
Buboes.
Right, often with fever, chills, headache, muscle aches, and extreme weakness.
And if it's not treated, it can become septicemic plague, right?
Unfortunately, yes.
Which is much more serious.
It is. Septicemic plague happens when those bacteria spread from the lymph nodes into the bloodstream.
And then,
It can lead to organ failure, shock, and even death.
wow, it's scary how something so small can cause so much damage throughout the body.
And then there's pneumonic plague.
Even more worrying because it's contagious, right?
Exactly. Pneumonic plague affects the lungs and can spread through the air.
Through respiratory droplets.
Right, and it comes on really fast with fever, chills, cough, chest pain, and trouble breathing.
So it's particularly dangerous because it can spread so easily from person to person.
That's right.
Okay, so we have 3 forms of plague, each with its own challenges.
And for all of them, prompt diagnosis and treatment are absolutely essential.
Early intervention is key,
I
right?
couldn't agree more.
So moving on to Y. pseudotuberculosis, It presents in a way that can be quite misleading.
Oh, you mean pseudoappendicitis?
Exactly. It mimics appendicitis, so you can get misdiagnosed and even end up having unnecessary surgery.
It can be a tough one to diagnose for sure.
Aside from that abdominal pain, what else does it cause?
Well, Y pseudotuberculosis can also cause fever, diarrhea, vomiting, headache, and joint pain.
It's like a chameleon, mimicking all sorts of different conditions.
And just like those other Yersinia, it can lead to complications like septicemia.
Right, so even though it might not be as infamous as Y. pestis, it's still not something to be ignored.
Definitely not.
Now, what about Y. enterocolitica, the one we associate with foodborne outbreaks?
Especially those linked to pork products.
Right, because it's often found in contaminated pork and it can survive in the fridge.
Makes it a real challenge for food safety.
So handling and cooking food properly is crucial to prevent those Y. enterocolitical infections.
Absolutely. Those precautions really matter.
But even when we're careful, people can still get infected.
It happens.
What are the typical symptoms then for Y. enterocolitica?
It depends on the person's age and health, but in children, diarrhea is often the most prominent symptom.
What about fever and abdominal pain?
Those two. And the diarrhea can last for weeks.
And in adults.
They're more likely to have abdominal pain and fever as the main symptoms.
And like with Y. pseudotuberculosis, that pain can be in the lower right abdomen.
Mimicking appendicitis again?
So we have two Yersinia species that can mimic appendicitis. Something to keep in mind?
Absolutely. Highlights the importance of a thorough evaluation and lab tests to make the right diagnosis.
Okay, so 3 distinct Yersinia species, each with its own set of clinical manifestations and possible complications.
It's important to know the ins and outs of each infection for effective management.
Before we jump into treatment, though, let's talk about how we actually diagnose these infections.
Good point. Laboratory diagnosis is essential.
Especially to make sure we get the right treatment.
Right. Let's start with plague, where a quick diagnosis is critical.
Especially in areas where it's endemic.
Exactly. So what's the fastest way to diagnose plague?
I'm not sure.
One of the fastest and most reliable methods is F1 antigen detection.
What's that?
It uses antibodies that specifically target the F1 antigen, which is only found on Y. pestis.
Like a bacterial fingerprint.
Exactly. And there are a couple of ways to detect it.
Like what?
One common technique is immunofluorescence microscopy.
Immunofluorescence microscopy.
Yeah, the antibodies are tagged with a fluorescent dye, so you can see the bacteria under a microscope.
So it's like highlighting them with a spotlight. Are there any other quick tests for plague?
There's also a dipstick test, even faster, and you can use it in the field.
It's great for remote areas or during outbreaks where a quick diagnosis is vital.
Exactly.
What about PCR? I'd imagine that's useful too for detecting Y. pestis.
Absolutely. PCR is very sensitive and specific.
It works how?
It amplifies specific DNA sequences from the bacteria, even tiny amounts.
So you can confirm a suspected case? Or maybe even detect it before symptoms show up.
It's a very powerful tool.
But even with those molecular methods, you still need to culture the bacteria.
Yeah, you do. Culturing gives you essential information.
Like what?
You can confirm the diagnosis for sure, identify the specific strain, and test its susceptibility to different antibiotics.
So it helps you choose the right treatment.
Exactly. It's like getting a full profile of your enemy.
But culturing Y. pestis is risky, right?
It is. It's classified as a biosafety level 3 pathogen.
Meaning?
You need special procedures and containment facilities to prevent accidental infection.
Not something you do in a regular lab.
Definitely not. Now, let's talk about diagnosing yersiniosis, those infections from Y. pseudotuberculosis and Y. enterocolitica.
I imagine culturing is still important, but are there any challenges?
Culturing these bacteria can be a little trickier.
How so?
They need specific media and conditions to grow properly.
For example?
When enterocolitica often needs a special medium called CIN agar and a slightly cooler temperature.
So more specific than just a standard agar plate.
Right. And for both Y. enterocolitica and Y. pseudotuberculosis, something called cold enrichment can be helpful.
Cold enrichment, what's that?
You incubate the sample at a very low temperature, around 4 degrees Celsius, for a few weeks.
And why do you do that?
It allows Yersinia to outgrow other bacteria in the sample.
Oh, so it's like giving them a head start.
Exactly.
Does PCR play a role in diagnosing yersiniosis?
Yes, it's becoming increasingly important, especially for Y. enterocolitica in food samples.
What about serology? Any role for antibody testing?
Serology can show past infections, but it's not so good for acute ones.
Why is that?
Because it takes time for the body to make antibodies.
So it's not the main diagnostic tool.
Right, so we've got traditional and molecular methods for diagnosing these infections.
But before we talk about treatment, it's important to understand how these bacteria spread.
You're absolutely right. Understanding the epidemiology of Yersinia is key for prevention and control.
So let's start with Y. pestis again, that plague agent.
As we said before, it's primarily zoonotic, meaning it spreads from animals to humans.
And the main reservoir.
Rodents, especially rats.
And fleas are the vectors, right?
They pick up the bacteria when they feed on infected rodents and then transmit it to humans through bites.
So controlling rodent populations and flea infestations is really important.
Absolutely. It's about breaking that chain of transmission.
And where is plague still a problem?
It's most common in Africa, Asia, and South America.
There are two types, right?
Yes, sylvatic plague and urban plague.
Sylvatic.
It's in wild rodent populations, while urban plague is in cities where rats are more common.
So urban plague is more likely to affect people.
And it's becoming more common as cities expand into areas where sylvatic plague is found.
So it's an ongoing problem, especially in places that are urbanizing quickly.
It is. Are there any other ways to prevent plague besides controlling rodents and fleas?
I'm not sure. Are there?
There are vaccines, but they're not widely used.
Why not?
They don't provide long-lasting immunity and can have side effects.
So they're mainly used for people at high risk.
Like researchers working with Y. pestis or people living in endemic areas.
Okay, what about Y. pseudotuberculosis and Y. enterocolitica? How do they spread?
They're both zoonotic, but unlike Y. pestis, they're often transmitted through the fecal-oral route.
Meaning?
Contaminated food and water are the main culprits.
So hygiene is really important.
Like washing your hands and cooking food properly.
Those basic food safety rules really matter.
They do. They can significantly reduce the risk of infection.
So tell me more about Y. pseudotuberculosis.
It's mostly found in animals, rodents, birds, and farm animals.
How do humans get infected?
Through contact with contaminated soil, water, or food.
And Y. enterocolitica?
It's more associated with pork products.
Why pork?
It survives at refrigeration temperatures, so it's a concern for the food industry.
Improper handling and storage of pork can easily lead to outbreaks.
That's right.
So the food industry needs to be vigilant about controlling the spread of Y. enterocolitica.
And consumers need to be aware of the risks of undercooked pork.
Public education is key here.
Absolutely.
And there are no vaccines for yersiniosis, right?
Not currently, so prevention really comes down to hygiene and food safety.
Okay, so we've covered a lot of ground, but now let's talk about treatment.
When it comes to plague, antibiotics are the first line of defense.
And acting quickly is essential.
Absolutely. The longer you wait, the higher the risk of complications.
What are the best antibiotics for plague?
Traditionally, streptomycin has been considered the gold standard.
But I imagine there are other options.
There are other aminoglycosides, like gentamicin, can also be used.
Why those?
They work in a similar way to streptomycin and often have fewer side effects.
What about tetracyclines, like doxycycline?
Those are another good option, especially for people who can't tolerate aminoglycosides.
How do they work?
They stop the bacteria from making proteins.
And what about fluoroquinolones, like ciprofloxacin?
They can be effective, but there's concern about resistance building up.
So they're not always the first choice.
They're usually saved for cases where other antibiotics aren't suitable.
So we have a few options for treating plague.
But it's crucial to choose the right one and be mindful of resistance.
Antibiotic stewardship is key.
Now, for yersiniosis, do you always need antibiotics?
That's a good question. It seems less aggressive than plague.
You're right. In many cases, yersiniosis is self-limiting.
So it resolves on its own.
Exactly. The body can often fight it off.
So just supportive care is enough then.
Things like rest, fluids, and over-the-counter medications for fever and pain.
But sometimes antibiotics are needed, right?
For severe cases, especially those with complications.
Like septicemia or reactive arthritis?
Exactly. In those cases, antibiotics are definitely recommended.
So what are the preferred antibiotics for yersiniosis?
It depends on the specific Yersinia species and how bad the infection is.
Give me some examples.
For Y. enterocolitica, trimethoprim sulfamethoxazole is a common choice.
Also known as?
Bactrim or Septra.
And are there others?
Fluoroquinolones like ciprofloxacin are often effective too.
And for Y. pseudotuberculosis?
Y. pseudotuberculosis is usually susceptible to ampicillin,
Which is?
a penicillin-type antibiotic.
Are there alternatives?
Trimethoprim sulfamethoxazole or a fluoroquinolone can be used too.
So we've got a good range of antibiotics to choose from.
But the choice depends on the situation.
Now, let's talk about managing these infections.
Well, as we discussed, supportive care is really important, especially for yersiniosis.
Making sure the patient stays hydrated, managing their pain and fever, and addressing other symptoms.
Exactly, those are the basics.
And for more severe cases,
hospitalization might be necessary for closer monitoring and intravenous fluids.
so it's about tailoring the management plan to the individual patient.
Exactly. And even after the acute infection is gone, follow-up is important.
Why is that?
Because some patients can have long-term complications.
Like what?
Things like reactive arthritis and post-infectious irritable bowel syndrome.
So those can stick around even after the Yersinia infection is gone.
It's important to manage those long-term issues, too.
So it's about providing care for both the short term and the long term.
Exactly.
Now, before we wrap up, how about we test our knowledge with those exam questions?
I love a good challenge. Fire away.
Okay, first question. What's that unique plasmid in Y. pestis that encodes the F1 antigen and helps fleas survive?
That's a good one.
Let me think. It's a P for a plasmid, right? Also known as PMT1.
That's right. It's like why pestis' VIP pass to the flea world.
Okay, hit me with question number 2.
Describe how the type 3 secretion system works and how it makes Yersinia so virulent.
Okay, here goes. It's basically a molecular syringe that injects proteins called YOPs into our cells. These YOPs then mess up our immune response, letting the bacteria survive.
Perfect. You really got the essence of how that system works and why it's so important for Yersinia.
2 down, 3 to go.
Next question. What clinical features tell why pseudotuberculosis and why enterocolitica infections apart, and what complications can each one cause?
Okay, so both can cause that pseudoappendicitis, mimicking appendicitis, but Y. enterocolitica is more likely to cause typical gastroenteritis symptoms like diarrhea. And for complications, Y. pseudotuberculosis can lead to septicemia, while Y. enterocolitica is more associated with reactive arthritis.
Fantastic job. You've got a good grasp of those key differences in their complications.
Putting on the next question.
Okay. What antibiotics are recommended for treating plague and yersiniosis, and why are some preferred over others?
For plague, streptomycin is the classic treatment, but gentamicin and tetracyclines are good alternatives. Fluoroquinolones can be used, but there's that concern about resistance. For yersiniosis, it depends. Many cases resolve on their own, but for severe cases, trimethoprim sulfamethoxyquinolones are good options for Y. enterocolitica, and ampicillin is usually effective for Y. pseudotuberculosis.
Another perfect answer. You've clearly been paying attention. You even remembered the resistance issue with fluoroquinolones.
Okay, last question. Bring it on.
What public health measures are important for controlling plague and yersiniosis, and how do they differ?
All right, for plague, it's all about preventing flea bites, so controlling rodents and using insecticides. There are vaccines, but they're not ideal. For yersiniosis, it's food safety and good hygiene, things like washing hands, cooking properly, and storing food safely. No vaccines for this one, so prevention really hinges on those practices.
You aced it. You really have a comprehensive understanding of Yersinia.
Well, I couldn't have done it without your help.
It's been a pleasure joining you on this deep dive. It's been a pleasure to be a part of this Yersinia deep dive with you.
Me too. It's amazing how much we've uncovered about this fascinating genus of bacteria.
There's always something new to learn in microbiology.
Definitely. Now that we've covered the general characteristics of the genus as a whole, I'd love to dig a little deeper into each species.
Sounds good.
Starting with Y. pestis, that notorious plague agent.
Oh, Y. pestis definitely stands out, even among the other Yersinia.
So what makes it so unique?
Well, as we mentioned earlier, it's a gram-negative coccobacillus.
Right.
And it displays something we call bipolar staining.
Meaning?
It stains more intensely at its poles, so it has this distinctive look under the microscope.
Interesting.
And unlike Y. pseudotuberculosis and Y. enterocolitica,
Which can move around.
right, Y. pestis is non-motile.
It relies on those flea taxis.
Exactly. It's traded in its flagella for a more luxurious mode of transport.
But what's really fascinating is its ability to survive in different environments and adapt to different temperatures.
We did touch on that.
It can hang out in fleas at cooler temperatures, but then it has to switch gears to thrive in the warm-blooded mammalian host.
That's quite a range.
It is.
And it manages this by carefully regulating its virulence factors.
Like how?
Well, for example, the F1 antigen.
That protective capsule.
Right. It's only produced at body temperature.
So it only puts on its armor when it enters the battlefield?
Exactly. And remember that protease we talked about?
The one that helps it spread?
That's also regulated by temperature, more active at body temperature.
So it can really spread from the initial infection site.
It's a master adapter, that's for sure.
It really is.
Yeah.
So let's revisit those different forms of plague for a moment,
Good idea.
because each one presents unique challenges.
Absolutely.
We've got bubonic plague, the most common form, with those painful, swollen lymph nodes.
Good buboes.
Right, along with fever, chills, and those other systemic symptoms.
And it's usually transmitted through flea bites, right?
It's like a tiny vampire injecting that nasty bacterium into our bloodstream.
When an infected flea bites, Y. pestis enters the bloodstream and travels to those nearby lymph nodes. It replicates there and causes inflammation, leading to those characteristic buboes.
And if it's not treated, bubonic plague can progress to septicemic plague,
Unfortunately.
which is much more serious.
Septicemic plague happens when the bacteria spread from the lymph nodes into the bloodstream.
Causing.
A systemic infection that can lead to organ failure, shock, even death.
It's a rapid escalation.
And then we have pneumonic plague, the most contagious form.
The one that's a real public health threat.
Exactly. Pneumonic plague affects the lungs and can be spread through the air.
When someone coughs or sneezes.
Right. That airborne transmission is what makes it so concerning.
Especially in crowded places where it can spread quickly.
Absolutely.
So 3 distinct forms of plague, each with its own set of clinical features and challenges.
And a rapid diagnosis is crucial for effective management.
We talked about some methods earlier, but what are the specifics?
Well, F1 antigen detection is one of the fastest and most reliable.
And we can use either immunofluorescence microscopy or that dipstick test.
Right. Immunofluorescence microscopy gives you that visual confirmation, while the dipstick test is a quick point-of-care solution.
Great for those field settings or during outbreaks.
Exactly.
But PCR also plays a role, right?
It does. PCR is very sensitive and specific.
It can amplify those specific DNA sequences.
Even from tiny amounts of Y. pestis.
So even if antigen detection is negative, PCR might still find it.
Yes, but even with these advanced methods, culturing is still important.
Why is that?
Culturing lets you isolate the specific strain of Y. pestis and test its susceptibility to different antibiotics.
So you know which treatment will work best.
Exactly.
So it's a combination of rapid tests for initial identification and culturing for those details about the strain and its antibiotic susceptibility.
It's a well-rounded approach for sure.
Now, what about treatment options for plague?
When it comes to plague, antibiotics are key, and acting fast is crucial.
We talked about streptomycin being that classic first-line treatment.
Right, but other aminoglycosides like gentamicin are also very effective.
And they often have fewer side effects.
That's a plus.
What about tetracyclines?
Those are another good option, especially if someone can't tolerate aminoglycosides.
And they work by
Inhibiting protein synthesis in the bacteria.
essentially stopping them from growing.
Exactly.
And fluoroquinolones, are they a possibility?
They can be effective, but we're seeing more resistance developing.
So they're not always the best choice.
It's better to reserve them for cases where other antibiotics aren't suitable.
Makes sense. So aside from antibiotics, what else is important for managing plague patients?
Supportive care is crucial.
Like what?
Intravenous fluids, pain medications, and oxygen therapy if needed.
And for those severe cases?
They might need intensive care.
So you're treating the infection and supporting the patient's overall well-being.
Exactly. It's a holistic approach.
Now let's move on to why pseudotuberculosis.
Another fascinating member of the Yersinia family.
It's a master of disguise, often mimicking appendicitis.
You're right, it's a tricky one.
So what do we need to know about this bacterium?
Well, as I mentioned, it's a gram-negative cucobacillus. And unlike Y. pestis, Y. pseudotuberculosis is motile at lower temperatures.
So it can move around in its environment?
Exactly. And it has a wider range of animal hosts, too.
Not just rodents like Y. pestis.
Right. It can infect a variety of mammals and birds.
Domestic and wild animals.
So it's a bit more of a social butterfly.
And probably more widespread geographically.
You're right. It's found on every continent except Antarctica.
So it's got a wider host range and a wider geographical distribution than Y. pestis.
It does.
Clinically, we know it can mimic appendicitis.
Pseudoappendicitis.
Right, but what other symptoms can it cause?
It can cause fever, diarrhea, vomiting, headaches, even joint pain.
Quite a range.
It can be difficult to pin down.
And like the other Yersiniae, it can lead to complications.
Yes, like septicemia.
So it can be serious.
In some cases, it can spread to the bloodstream,
Causative.
a life-threatening infection, and it can also infect other parts of the body.
Like where?
The liver, spleen, bones.
So even though most cases are mild, it can take a turn for the worse.
It's important to be aware of that potential.
How do we diagnose these infections in the lab?
Culturing is still the gold standard for Y pseudotuberculosis.
And what kind of samples?
Blood, stool, lymph node biopsies, pus from infected sites.
What media do you use?
It grows well on standard blood agar and McConkey agar.
And are there selective media?
CIN agar can be used to help isolate it from stool samples.
And temperature is important, right?
Absolutely. Y pseudotuberculosis prefers those cooler temperatures.
What about PCR?
PCR can be helpful, especially for detecting the bacteria in environmental samples or food.
So it helps us track it in different settings.
Exactly, and serological tests can show past infections.
But not so good for active ones.
Right, because it takes time for those antibodies to develop.
So serology might give you some clues, but it's not the primary method for diagnosis.
Now let's talk about treatment for Y pseudotuberculosis.
From what I remember, it's susceptible to a wide range of antibiotics.
You're right, it usually responds well to treatment.
So what are our options?
Ampicillin, amoxicillin clavulanate, trimethoprim sulfamethoxazole,
And
fluoroquinolones, third-generation cephalosporins.
so we have quite a few choices.
The best choice will depend on the specifics of the case.
And supportive care is still crucial, right?
Absolutely. Rest, hydration, pain management.
You treat the whole patient.
Exactly. A holistic approach.
Now let's turn our attention to Y enterocolitica,
The one often linked to foodborne outbreaks.
especially with pork products,
It's commonly found in pigs' intestines.
and can contaminate pork during processing.
That's the main route of transmission to humans.
Eating undercooked or contaminated pork.
But it can also spread through water or contact with animals.
So hygiene is important.
Absolutely, in all aspects of food prep and animal handling.
But even with precautions, infections can still happen.
It's true.
So what are the typical symptoms of a Y enterocolitica infection?
It can vary depending on the person.
How so?
In children, diarrhea is common, along with fever and abdominal pain.
And it can last for weeks.
In adults, abdominal pain is often the main symptom.
And it can mimic appendicitis.
Right, that's right, which makes diagnosis tricky.
So we had to be careful.
Adults might also have fever, diarrhea, nausea, vomiting, headache.
And those long-term complications like reactive arthritis.
That's one of the more concerning complications.
It can be quite debilitating.
And there's also erythema nodosum.
Those painful red nodules.
Yes, although it's not as common as reactive arthritis.
So even though most infections are mild, those complications can have a big impact.
Early recognition and management are key.
How do we diagnose these infections?
Culturing is still the best way to diagnose Y enterocolitica.
From what samples?
Stool, blood, biopsies, pus.
Any specific media?
Blood agar, McConkey agar, and CIN agar can be used.
And temperature.
Remember why enterocolitica likes it cool, 25 to 30 degrees Celsius.
So similar to Y pseudotuberculosis, but a bit cooler.
Right.
What about PCR?
PCR is very useful, especially for food and environmental samples.
So it helps track the bacteria in different settings,
And serology can detect past infections,
but not so good for acute ones.
because antibody production takes time.
Okay, so what about treatment for Y enterocolitica?
Y enterocolitica is usually sensitive to antibiotics, which is good news.
So what are our options?
Trimethoprim, sulfamethoxazole, fluoroquinolones,
Anything else?
third-generation cephalosporins, aminoglycosides.
So we have several choices,
The best one depends on the specific situation.
and supportive care is essential to that.
Absolutely. Rest, hydration, pain management, sometimes hospitalization.
So a multi-pronged approach.
Exactly.
Now, before we wrap up, let's talk about prevention, especially for Y enterocolitica.
It's a major cause of foodborne illness, so prevention is key.
How can we prevent it?
The food industry has a big role to play, with hygiene practices during processing, thorough cooking, and proper storage.
So it's up to them to minimize contamination.
But consumers can also take steps to protect themselves.
Like what?
Cooking pork thoroughly, washing hands after handling raw meat, avoiding cross-contamination in the kitchen.
So it's a shared responsibility.
It takes everyone working together.
Well, I think we've covered all the essential aspects of Yersinia.
It's been a really interesting deep dive.
I've learned so much.
It's amazing how these tiny organisms can have such a big impact on human health.
It really is.
Knowledge is power when it comes to these infections.
Absolutely. Understanding them helps us diagnose, treat, and prevent them.
Thanks for joining us on this journey into the world of Yersinia.
And to our listeners, keep exploring the fascinating world of microbiology.